Welcome to part 3 of this webinar series on systematic reviews and meta-analyses. In part 1, I reviewed what SRMAs are, their purpose, and the importance of combining a systematic review with a meta-analysis. In part 2, I reviewed the steps in conducting a systematic review, and here in part 3, we will review the steps in the conduct of the meta-analysis. My only disclosure is that I am currently a MyTax Elevate postdoctoral fellow jointly funded by the Government of Canada and the Canadian Sugar Institute. Now let's consider, what is a meta-analysis? A meta-analysis is a statistical technique for combining the findings from several independent studies to resolve discrepancies and provide a quantitative estimate of the treatment effect. It gives a larger sample size than any individual study, allowing for greater precision of estimates as well as identifying sources of diversity. Here are the steps in performing the meta-analysis, each of which comes with a lengthy description, but I will just mention them briefly in the interest of time. Again, as with the general steps I reviewed for the conduct of the systematic review, by understanding these steps in the conduct of a meta-analysis, they can help you to critically assess SRMAs, and we'll review them again in a subsequent video. Meta-analyses can be performed in accordance to guidelines outlined in the handbook put together by the Cochrane collaboration. It describes how to use certain software in order to enter data and obtain a quantitative estimate of the overall effect. The handbook is available online at the link presented here. Note that there are various statistical software packages available which can be used to run the meta-analysis. STATA is one example that is commonly used. This is an example of a forest plot generated by STATA using the data extracted from clinical trials. This data uploaded into the STATA program includes each study, the sample size, and the extracted outcome data and variability around it. In this figure, the extracted data include mean differences and standard errors, which are then converted by STATA to 95% confidence intervals. STATA uses this data to create a forest plot, seen here in the middle, as well as calculating and presenting the weights of each study using the variance, and then the summary estimate of the effect for the pooled data, which we see here as the green diamond at the bottom. This green diamond represents the overall effect of all included studies. One additional note with regard to the column on the weights of each study, you'll see how they are not all exactly the same for each study. A meta-analysis is not a simple average of the studies, but the studies contribute more or less weight depending upon their precision, which we represent using the variance around the mean effect for that study. This is called inverse variance weighting. So a study with a small variance, which means it's more precise and it's generally a larger study, will get a larger weight in the overall calculated summary estimate compared to a study with a large variance, which is less precise and is usually a smaller study. The data used in a SRMA depends on the type of studies, clinical trials versus observational studies. Clinical trials have mean differences, which if you are pooling data using different units that cannot be converted to the same unit, you then use standardized mean differences. This is in contrast to observational studies, which usually report relative risks. To keep things simple, I will use examples of clinical trials and thus mean differences in the rest of this presentation. Here I have a very simple forest plot to review how to read it. In number 1, you can see we first have an indicator of the study included. In this case, we have the author of the publication. Sometimes SRMAs may use author, et al, and year. Jumping to number 3 here, this is the mean difference and 95% confidence interval around the endpoint for that study. So the effect found within that one study. Then for number 2, we have the weight of the study in the analysis. This is given by the meta-analysis software based on the variability around the endpoint as you have entered it. If there is high variability, it will have a lower weight in the analysis, meaning that its contribution to the overall effect estimate will be lower because it is less precise compared to a study with low variance. To interpret the forest plot, first take a look at the scale at the bottom. The 0 means that there is no difference in outcome whether you are given the intervention or the control. To the left means that there is a reduction, and to the right means that there is an increase in the outcome when given the intervention compared to the control. So if we were talking about body weight as the outcome, then the first study in green here shows a reduction in body weight when given the intervention compared to the control. Thus, this means that the study shows a result which favors the intervention. Conversely, the second study in blue shows that there is an increase in body weight when given the intervention compared to the control, which means that it demonstrates a favoring of the control in terms of body weight. The diamond at the bottom of the forest plot is the overall pooled estimate of the effect of the intervention compared to the control. The left and right sides of the diamond are the 95% confidence intervals of the pooled estimate. This is the overall effect resulting from the pooling of all the studies. Now let's explore an important term in meta-analyses, heterogeneity. Heterogeneity is a statistical test done which assesses whether observed differences in results between studies are the result of chance alone, or if they are the result of variability in the included studies themselves. So beyond differences in the results between studies that would result from chance, studies may have clinical differences, including the type of participants included, the way the intervention was administered, and the way the outcomes were assessed. Studies may also have methodological differences, including how studies were executed. For example, if they were crossover or parallel in design, so in the methods of the actual structure of the study. Here's the classical example of heterogeneity. We have two studies with opposite results, one showing an increase and one showing a reduction as a result of the intervention. Heterogeneity here is 98%. This is found beside the I-squared, which is the outcome of the statistical test, along with the p-value that we would want to look for in the forest plot to see what the heterogeneity of the analysis is. So an I-squared of 98% and a p-value less than 0.001 means that the variability in the included studies is most likely not due to chance, but due to diversity between the studies themselves. This quote explains it with a simple example. If you have one hand in the fireplace and the other in the freezer, on average, you're just fine. This example demonstrates that the actual conclusion in the example should be that the results of the two studies are likely due to very different environments or study designs, and probably not just due to chance. This is why it is important to have subgroup analyses to explore whether there are significant differences between the design aspects of the included studies, which may be modifying the effect on the outcome variable. These are the general guidelines for assessing heterogeneity. So anything below an I-squared of 30 to 40% may not be important, but anything greater than 50% is considered substantial. So after we have detected heterogeneity and quantified it, we now need to try to address it. To deal with heterogeneity, we can do sensitivity analyses, in which we remove one study at a time to see if it alters the heterogeneity, that is, to see if removing one study reduces heterogeneity so that it is no longer significant. If you find that by removing one study, heterogeneity goes from being significant to not significant, there may be something different about this one study compared to the others, which is significantly modifying the effect on the outcome. We can also conduct the previously discussed a priori subgroup analyses to explain the heterogeneity. Thinking back to one of the previous videos, we went over an example of Dr. Black and Dr. White. In the overall analysis, the I-squared is substantial at 83% and is significant. However, let's say we did an a priori subgroup analysis by population type and saw that in the first set of studies that those were conducted all in participants that had cancer. And in the second set of studies, we had populations free of chronic disease. If, let's say, the test for the subgroup by population type was significant and the I-squared within each subgroup was at 0%, we may conclude that the heterogeneity of the overall analysis may be explained by population. Here's an example from a SRMA done by our group, which helps to demonstrate the importance of subgroup analyses. What is important to know in this context is that it was determined a priori to conduct sex-specific subgroup analyses because of possible differences in effect between men and women on risk of coronary heart disease. The overall effect, which is depicted by the diamond right at the bottom of the forest plot, demonstrates that with all studies taken together, there is a significant effect of increased risk of CHD. The heterogeneity is significant with an I-squared of 43%, which I've highlighted in this purple box here, and it's considered moderate variability. However, when the subgroups are separated based on sex, there is a significant difference between the effects in either group. This is presented at the bottom of the plot under test for subgroup differences, and the p equals 0.02. If we look at the groups individually for women at the top, there is a significant effect. However, for men, there is no significant effect. Therefore, the conclusions for this SRMA would be that there may be no effect in men. However, there may be a significant effect in women. This demonstrates the importance of conducting subgroup analyses and the influence it can have on the overall conclusions and interpretation of the results. Here's an example of a set of a priori subgroup analyses performed in another meta-analysis to investigate any effect modifications. This is just to give you an idea of one of the ways multiple subgroup analyses can be presented. Note there are subgroups representing potential clinical differences between studies, such as baseline LDL cholesterol levels, and methodological differences like study design and duration of the intervention. The p-value to assess if there is an effect modification for each subgroup is in the column farthest to the left. Note that these figures are typically always in the supplement, but described in the results. The previous slide presented results for categorical subgroup analyses. For data that is continuous in nature, like dose of the intervention, or follow-up of the duration of the intervention, and baseline outcome level, continuous subgroup analyses can be performed, where an example is presented here. Each of these can also be presented graphically, so you can see the linear, if there is a linear effect. For example, for baseline LDL cholesterol in this analysis, there is a significant negative association. Whereas baseline LDL cholesterol increases, a greater reduction in the outcome of LDL is observed in these studies. Another important part of analyses include dose-response analyses when possible. And these include both linear and non-linear dose-response analyses. The example here demonstrates a significant linear dose-response effect, seen by the solid red line, of the consumption of fruit on incident hypertension in cohort studies. there was no significant nonlinear effect as depicted by the black line and dotted lines, which represent the 95% confidence. It is important to understand the quality of your data. High quality data, which is low in bias, is closer to the truth, which we're trying to determine. Conversely, lower quality data is high in bias and thus deviates from the truth, so should be interpreted more cautiously. There are two areas in a SRMA where risk of bias should be assessed. One is the assessment done on each individual study included in the SRMA to assess the study quality. As discussed earlier, this can be done using assessment tools such as the Cochrane Risk of Bias tool for trials. As a reminder from a previous presentation on extracting data for risk of bias, this includes multiple categories in which risk of bias occurs. Examples include the categories of sequence generation and blinding, which is assessed individually for each study and then rated as either low risk of bias, high risk of bias, or unclear. And depending on the tool that you are using, it can vary in the degree of these categories. The most recent ROB2 tool has additional classifications. Here is an example of how risk of bias assessments can be reported in SRMAs, where if you see a lot of red, this means that there are a lot of studies with a high risk of bias in certain domains. This is important to review and note that if there is a lot of studies rated as having a high risk of bias, this should be stated as a limitation and bring caution to the interpretation of the conclusions due to low quality of the data. Risk of bias assessments can also be used to complete subgroup analyses, where you can compare those studies at high risk of bias versus those at low or uncertain risk of bias to see if the results differ significantly and whether this affects interpretation. The second area in a SRMA where risk of bias should be assessed is to assess if there is any bias which can result from differences in studies which are not published, which we call publication bias. This assessment is performed at the end of the meta-analysis. To assess publication bias, you first create a funnel plot of the studies to assess for asymmetry, and this is done using the statistical software package. As a rule of thumb, tests for funnel plot asymmetry should be used only when there are at least 10 studies included in the meta-analysis, because when there are fewer studies, the power of the test is too low to distinguish chance from real asymmetry. The funnel plot is a plot of each study by their effect and variance, and the solid line represents the overall pooled estimate of the effect. To assess for publication bias, you can look at the funnel plot to visually inspect for asymmetry. You can also perform statistical tests for publication bias, known as the Eggers and Beggs tests. Note that what this may mean is that there are small studies typically favoring the control that are missing, meaning that they have not been published. This is why it's important to assess for publication bias, and if it is detected, to use methods to explore it. To address publication bias, you can perform trim and fill analyses in which those areas in which there are missing studies, they will be imputed, and then the pooled estimate of the effect is recalculated. To assess whether there is a significant small study effect, we compare the recalculated pooled effect to the original pooled effect. Has the direction of the effect changed, or has the significance of the effect changed? If the recalculated pooled effect would give you a different conclusion, then there is a significant small study effect, and this is an important limitation to the SRMA and needs to be acknowledged. This will reduce the confidence you have in the main SRMA results. The final step in performing a SRMA is to do an assessment of the overall certainty in the evidence. And this can be done using the grade approach. We will review grade in the next video. So that pretty much wraps up these steps in the conduct of the meta-analysis, and I hope you found them informative and useful. I'd like to acknowledge my mentors and lab group, as many of these slides have been developed and shared by members of our team.